We are going to see kinetics of enzyme catalyzed reactions. That means how different factors are affecting the activity of an enzyme. Look at these two names, Michaelis and Menten. They have contributed a lot towards the development of this branch of biochemistry. What are the different factors that affect the activity of an enzyme? For that, first of all, we should know what do we mean by the term activity, enzyme activity. It is the speed of an enzyme. How many moles of the substrate are converted into product in unit time? Or how many moles of product are formed in unit time? That is what is called enzyme activity. Usually, temperature, pH, and concentration of substrate are the three major factors that affect activity of the enzyme. Apart from that, presence or absence of any inhibitor and sometimes salt concentration are the other factors that may affect the activity of an enzyme. What are the different factors that affect? How do we study the effect of concentration of substrate upon the activity of the enzyme? When we complete our study, we get a graph like this. Remember, the graph has two axes, x and y. What is written on the horizontal x-axis? It is the concentration of the substrate. The unit is millimoles. You can see 0, 0.5, 1, 1.5 like that. On the y-axis, we have the velocity. The unit given there is micromoles per minute, which means how many micromoles of the substrate are converted into product per minute. That is what is given there. And there you should understand that what we measure is the initial velocity. And how do we do it? We take the series of test tubes, fill them with all the raw materials and at the necessary pH, etc. Enzyme is added. Same concentration of enzyme is added to all the test tubes, and we incubate all the test tubes at a particular temperature. We say optimum temperature and pH for a definite time period. Time period may be 10 minutes, 15 minutes, 20 minutes like that. At the end of this incubation time, we measure the concentration of the product. And how do we measure? A colorimeter in our lab is sufficient. So we measure the concentration of the product and we mark it in the graph paper. You see, in this graph paper, the first test tube has a concentration, say just after 0. It may be around 0.1 like that. Then we have 0.2. Then third one is around 0.25 like that. And when the substrate concentration is 0.5, you can find out from the graph what is the corresponding velocity. It is around 7. And when the substrate concentration is 1, the corresponding velocity is just more than 10. It is around say 11. When the substrate concentration is 2 millimole, the velocity of the reaction is just about 12 micromole per minute. So like that we get a graph. So we plot a graph with the substrate concentration on the x-axis and velocity on the y-axis, and we get a particular shape. This shape is called rectangular hyperbola. This is a typical Michaelis-Menten kinetics. Michaelis and Menten, two wonderful scientists. Their picture is given in the introductory slide. A typical Michaelis-Menten plot is shown here. You can see it's a rectangular hyperbola. The graph has typically two regions. In the initial region, you can see on the x-axis the substrate concentration is increasing, and correspondingly the enzyme activity, the velocity also is increasing on the y-axis. So the graph is curving towards the right. It's because when the enzyme concentration is lower compared with the active sites of the enzymes, as we add more of the substrate, more of the enzymes are able to do their work. Less enzymes are sitting lazy. And then as we are adding more of the substrate, a time moment will come when all the enzymes are doing their maximum work, or there are sufficient substrate molecules so that the active sites of all the enzymes are filled up. Then we can say the enzymes are all saturated with the substrate. Thereafter, even if we add more of the substrate, the velocity, the enzyme activity does not increase correspondingly, and we can see a horizontal region for the graph. It's because already all the enzymes are doing their maximum activity, and even if we add more of the substrate, they need to wait till the active sites are free to take up the extra substrate molecules we added. This is the typical Michaelis-Menten plot. And you can see there are maximum rate, Vmax. As is shown in the graph, we cannot exactly measure the Vmax by using such a graph. This graph is showing a certain line for a Vmax, but that may not be correct always. As I told you earlier, initially as the concentration of the substrate increases, velocity of the reaction also increases. There we say the velocity is determined by the concentration of the substrate. It is a first order kinetics. Velocity depends upon the concentration of substrate. Thereafter, once all the enzymes are doing their maximum work, once all the active sites are filled up, then the rate does not depend upon the concentration of substrate. We say it is zero order kinetics. In this graph, you can see a term Km value. It is on the x-axis. You know that on the x-axis what is plotted is substrate concentration. So there is a relation between substrate concentration and the Km value. What's it? Can you find out the corresponding velocity? It is given as 1 by 2 Vmax or Vmax by 2. It is half the value of maximum velocity. From the graph we know what is the Vmax, and just half of the Vmax is 1 by 2 Vmax. The corresponding substrate concentration is called Km value or mm constant. Km value is of particular importance. What is the significance of Km value? Often we say Km value is a signature of an enzyme, and it is particularly constant for a given enzyme. Km value is actually the concentration of the substrate at which half of the active sites of the enzyme in the given medium are filled. And it provides us a measure of the substrate concentration required for significant catalysis to occur if we are running a laboratory in which enzymatic reactions are carried out to produce a product. We should know the Km value of the enzyme. A higher Km value indicates that more of the substrates is required for a significant catalysis to occur. And a low Km value indicates that even at a lower concentration of the substrate, significant catalysis can occur and product can be produced. So, Km value is a measure of the ability of the enzyme towards the substrate. If the Km value is lower, the enzyme has more affinity towards the substrate and more reaction can take place at a lower concentration of the substrate. What's the significance of Km value? It is constant for a particular enzyme under a given set of conditions. And these Km values are used in practical works in enzyme assay, etc. to identify how much is the activity of an enzyme. And the Km value and Vmax can be affected by different conditions, for example, pH, temperature, and other factors. Km denotes the enzyme affinity for its substrate. The higher the Km value, the lower the enzyme affinity for its substrate. And therefore, more of the enzyme is required to carry out the activity at a particular rate. Km permits evaluation of the inhibitor type. We will see the slide later. Isoenzymes, different forms of the same enzymes catalyzing the same biochemical reactions, differ in their Km value. During the course of an enzymatic reaction, as you can see in the equation, the enzyme and the substrate come together with a constant of K1, as you can see in the equation, and an enzyme substrate complex is formed. The enzyme substrate complex has two fates. It may either be converted back into free enzyme and substrate. You can see the constant is K minus 1. Or the enzyme substrate complex may be converted into enzyme and product. That means the substrate is converted into product and the product may go away from the enzyme. In that case, the enzyme is again free to accept another substrate. You know that this phase is a slow phase compared to the initial phase. And accordingly, we can give rise to the Michaelis-Menten equation. V0, what is V0? V0 is the initial reaction velocity which we used to plot the graph, is equal to Vmax. What is the Vmax? Vmax is the maximum velocity we observed and into concentration of substrate. Yes, with the square bracket indicates concentration of substrate divided by Km. What is Km value? It is the concentration of the substrate at which velocity of the reaction is half of the maximum velocity plus concentration of substrate. This is the valuable world famous Michaelis-Menten equation. What's the effect of temperature upon an enzymatic reaction? You can see in the graph, when the temperature increases from 0, the rate of the reaction is slowly increasing, and in this picture around 40-41 degrees Celsius the rate is reaching the highest value. Then any further increase in temperature sharply reduces enzyme activity. Here the enzyme activity doubles with every rise of temperature by 10 degrees Celsius, because you know, as the temperature increases, the substrate molecules are colliding faster, they are getting more energy, they are colliding faster and they can take part in the chemical reaction. But you know that most of the enzymes are proteins and a higher temperature denatures, destroys the protein structure. Therefore, once the optimum temperature is achieved, which means the highest reaction rate at a particular temperature, once that temperature is achieved, thereafter any further increase in temperature will denature the enzyme, therefore the activity decreases. What do you mean by the term optimum temperature? It is the temperature at which the enzyme is demonstrating highest activity. Below the optimum temperature, activity decreases because the enzyme molecules become inactive and the substrate molecules have less amount of energy. While after the optimum temperature, the reaction rate is suddenly decreasing because of the heat-induced denaturation of the enzyme molecules. This is typically applicable for any enzyme. Of course, the optimum temperature may be different for different enzymes. Enzymes of animals may have a higher optimum temperature, while enzymes of plants may have a lower optimum temperature. Why we are using a refrigerator in your home to keep food materials without getting damaged? Because in the lower temperature of the refrigerator, enzymes of the decomposing microbes are not able to function. So we are using this feature in our day-to-day life. What's the effect of pH, changing pH upon the activity of an enzyme? As you can see in the graph, from the beginning, an increase in the pH leads to an increased activity of the enzyme. Then the activity reaches a maximum value at a pH called optimum pH. Thereafter, any further increase in pH reduces the activity of the enzyme. You know that pH is a measurement of the concentration of hydrogen ions. And as the pH varies, ionization of the enzyme molecules take place there actually. Since enzymes are proteins made up of amino acids, these amino acids are having side chains. They may have a different degree of ionization at a different pH. So at any given pH, the enzyme has a particular activity, and as the pH increases, the activity also increases to a certain level. Thereafter it decreases. Why we are using a lowered pH in order to preserve food materials in the form of pickles? What's the biochemistry behind pickling of food materials? Different enzymes have different values of optimum pH. Chymotrypsin is a protein digesting enzyme in our alimentary canal. What is optimum pH for chymotrypsin from the graph? It's around 8. While for choline esterase, the value of optimum pH is slightly around 8, we can say. What about for pepsin? Pepsin is the protein digesting enzyme in our stomach, and its optimum pH is around 2. You know that in our stomach, acidic pH is created by hydrochloric acid. Then you have papain here in the diagram. Papain is another enzyme which we obtain from papaya, etc. There, after pH 4, activity is steady. So different enzymes have different values and different shapes of the graph for optimum pH. Here is a Lineweaver-Burk plot. It is also called double reciprocal plot. What's it? It is obtained from the Michaelis-Menten plot, which we found earlier. To create an LB plot, Lineweaver-Burk plot, we obtain reciprocal values of velocity and substrate concentration. Here on the x-axis you can see 1 by S, which means reciprocal of the substrate concentration on the x-axis. On the y-axis you can see 1 by V, which means reciprocal of the velocity values we obtained earlier. And we get a plot. Why do we do it? In the earlier rectangular hyperbola plot of the equation, I told you it is very difficult to pinpoint Vmax exactly. Since it is a hyperbola, we cannot exactly measure the Vmax. There is the application of this LB plot. You can see here the graph is a straight line, it is intercepting the y-axis. That point is called 1 by Vmax. It is if we plot it backward, it will intercept the x-axis. That point is called minus 1 by Km. So here we can exactly obtain the values of 1 by Vmax and minus 1 by Km. And once we have 1 by Vmax, you get the reciprocal of that value, we have the Vmax. So the advantage of LB plot is that we can get exact values for Vmax and Km. And this LB plot has one more importance. Presence of any type of inhibitor in the reaction medium can be identified from this LB plot. How do we do it? We will see when we come to enzyme inhibition. Here is an assignment for you. A small work. A scientist has done an enzyme work and he got the values of substrate concentration and velocity. You have two duties. Duty 1. You take a graph paper, plot the substrate concentration on the x-axis, velocity on the y-axis, and you get the graph. Duty 2. You get the reciprocals of these values using a calculator. You have your mobile phone. In your mobile phone, you have the calculator. You get the calculator. You type 1 by 2.5. You get the reciprocal of 2.5. You type 1 by 5.0. You get reciprocal of that one. Like that, you get the substrate concentration reciprocal for all values. You get the 1 by V values for all this data given, and you plot the graph. It will become an LB plot. And once you do it on the graph paper, you draw the final graph using a pen, get a photograph, and send it to me as you give me the result of your examination and also the answer. Wish you all the best.